Thank you for inviting me to present my research in the EASF webinar. And my background is in neuroimaging. So the topic I'm going to present today is how we can leverage neuroimaging techniques to extract neural and physiological dynamics. So hi, just give a very nice introduction of myself, but I also have some brief slides on it. I received my bachelor's degree in China, like Jingyuan University, and my undergrad degree was in biomedical engineering. And later, I pursued my PhD training at Stanford Radiology Science Lab, focusing on signal processing, statistics, and general neuroimaging methodology. And later, I moved to Boston for postdoc training in high-field functional magnetic resonance imaging and also multimodal fusion. I'm currently an instructor and at the Martinos Center and will start my own lab as an assistant professor in the coming months. So my research generally can be summarized by two themes. One theme is to advance general neuroimaging methodology. So we know that there are many techniques to look at the various aspects of our brain, such as the EEG to look at the scalp electrophysiology, and functional magnetic resonance imaging to look at the whole brain hemodynamic changes, and the PET, positron emission tomography to look at the neurochemical processes of the brain. So along this direction, one focus is how can we achieve the optimal usage of different neuroimaging techniques. So what are the spatiotemporal properties and biophysical limits of these different neuroimaging modalities? And also, what's the best practice to acquire and analyze the data in order to maximize the sensitivity and neuronal specificity of each modality? And on the other hand, these different techniques are very complementary in nature. For instance, for fMRI, it can have access to deep brain structures, have whole brain coverage, and it has excellent sensitivity, but it also have the downside of being not a kind of modest neuronal specificity. Basically, it's an indirect measure of neural activity, which I will introduce in later slides. And also, it wouldn't have access to a broad array of molecular processes that are key to brain's function or dysfunction. But for EEG, the scalp EEG electrode measures, they can offer direct measures of neural activity. That's kind of complementary to fMRI. And for positron emission tomography, it's an excellent tool to image these molecular processes non-invasively in human. And not just for their mechanisms, if you look at their spatial and temporal scales, they are also very complementary to each other. So my research also focuses on how can we combine these different neuroimaging techniques to address the limitation of each modality, and how can we integrate the rich information gathered from different modalities to achieve a holistic view on the brain's function and physiology. So as I mentioned, one focus is to advance these techniques, but I also have strong interest in applying these techniques to study the brain intrinsic functional and physiological dynamics. So first is what is intrinsic brain dynamics? So if you inspect the raw fMRI time course during the resting state condition, meaning the subject not doing any task, just simply inspect its time course over time, you can already see there are lots of ongoing dynamics there. As I will introduce later, they have very organized patterns in this rich intrinsic brain dynamics. So on one hand, it's not quite surprising because in this default resting state, your brain is being comprises of different levels of attention and maintaining consciousness and processing information for interpreting, responding to, and predicting environmental demands. And the functional architecture of these brain dynamics can also be altered by the arousal levels. For instance, sometimes you're alert and sometimes you're a bit drowsy, or it can be disturbed by external stimuli, or when you kind of struggle with some life difficulties. So the questions I'm also interested in are what are the brain dynamics look across different brain conditions or disease conditions. And second is what are the mechanisms underlying these state transitions? And finally, whether by looking into the first two questions, we can decipher new biomarker opportunities for these various psychiatric or neurological disease. And the particular angle we're focusing on is really leveraging the recent advances in neuroimaging. So for instance, these include whether we can use ultra-high field functional magnetic resonance imaging, which allow finer scale depictions of those subcortical and brainstem regions and cortical layers to achieve fine-grained characterizations of whole brain functional architecture. And whether we can combine multimodal imaging such as PET and EEG to help elucidate some physiological and neuronal changes seen in the functional magnetic resonance imaging. And whether we can combine in PET and fMRI to look into the energetic underpinnings of large-scale brain changes. And finally, we can probe whether how the brain functional dynamics change in response to a release of a specific neuromodulator. So that's kind of my general interest. So for my talk today, I will cover two threads of research, and for each thread, I kind of align with one theme. So for the first part of my talk, I will just share some explorations into the opportunities and pitfalls of fast functional magnetic resonance imaging. That's a primary modality I've been focusing on for quite a while. So first is what is functional magnetic resonance imaging? So its mechanism is actually very straightforward. So for instance, when you have an external stimuli and then your neurons are going to fire and go active, and it would need additional glucose and also oxygen delivered to the local region. But our brain is such a luxury device, it always delivers more oxygen than what is needed in the local region. So end up compared to the baseline condition during the activated state, there is increased oxygenated hemoglobin in its level and a reduction in deoxygenated hemoglobin. But because deoxygenated hemoglobin is paramagnetic, so the concentration of its change is going to alter the magnetic property that's going to be captured by the MRI scanner. So basically, when there is a stimulus, there will be elevated signals we can observe in the fMRI. So here is just like a simple summary of these cascading hemodynamic changes. So there is stimulus and then neurovascular coupling, and later there's elevated fMRI signals detected by the MRI machine. And usually you can, the red spot here kind of highlights which region have the increased response. But as you can see, this process is a bit sluggish. Typically, if you monitor the temporal course of the fMRI signals in the activated region, you can see it may peak really after several seconds after stimulus onset. So this process is kind of slow, so its major power of this fMRI signal is also dominated by ultra slow frequencies. So if you plot the spectrum, you can see the power is dominated by signals like with power below 0.1 Hertz. So that's a typical case where people believe that's the temporal limits of fMRI. And because this sampling rate, this temporal characteristic is super slow, so the sampling rate of fMRI is really on the 2 to 3 second scale for quite a long time. But recently there has been like MR engineering developments which allow us to acquire multiple brain slices at the same time. The direct consequences, like previously we can, for instance, take 2 seconds to acquire the whole brain image. But then now within the same duration, we can have multiple snapshots of the brain dynamics. So it's really pushed the temporal resolution of fMRI acquisition to much better regimes. So the question is whether, because vascular sampling comes the consequence, you will have elevated power at higher frequencies. For instance, when you change the sampling rate from 2 seconds to about 0.7 seconds, in addition to those better temporal precision, you will also see there's much lots of information in the higher frequency band got reviewed. So we call this kind of fast fMRI kind of beyond the 0.1 hertz here. So the question is like, are there signal or it's just pure noise revealed in the higher frequency fluctuations? This is a pretty interesting side because there are lots of interesting brain oscillations occurring at frequencies way above 0.1 hertz. For quite a while, people think it's impossible to observe it with fMRI. So there has been lots of studies started looking into these questions when we were able to sample data much faster. So for instance, this study looked at the brain functional activity patterns, just concentrating on those activated regions highlighted by red. You can see not just in the typical below 0.1 hertz range, in the 0.5 to 0.8 hertz range, you can also see the clear functional activity. These are doing devoid of any external stimuli. And also people characterize it using impulse stimuli. So here you have the oscillatory visual stimuli and characterize its response. In the primary visual cortex, as you can see, even up to 0.75 hertz, you can still see clear or significant fMRI response, although the magnitude is much lower compared to lower frequencies. So that's kind of a little bit contradictory to the conventional notion that hemodynamic response is quite slow. So the question we started to look at is like, are these higher frequency observations people are seeing really arise from those hemodynamic changes, just as low frequency signals as well? So one way we look at it, it can be based on the signal characteristics of MR acquisition. So typically, if the signal, if it arises from hemodynamic response, its percentage signal change should scale linearly as function of echo time. So echo time is a MR acquisition parameter you can set when you acquire the data. So in this case, if you can acquire multiple sessions of the data with different echo time, and it will open up a possibility to test whether a particular component you are seeing, whether it's from the hemodynamic mechanisms or not. So that's what we did. So we basically collect functional data as a function of different echo time, and here plot the results. They're quite similar in different parts of the brain shown on the left and right panel, but for instance, just concentrating on the left panel here. And the different colors here means different frequency. And you can see even up to the 0.4 to 0.5 hertz, which is the red curve shown here, it still show a linear dependence on echo time. So this basically suggesting these higher frequency phenomena people are seeing still arise from hemodynamic mechanisms. So the question next is why these hemodynamic response can be faster than the conventional characterization using strong stimuli. And then one possibility we were thinking is for instance, in a task-free condition, your brain is not, the neuronal activity is not in a constant strong level. If you look at the local field potential, you can see there is varying levels of neural activity throughout the entire scan and monkey acquisition. And then it's a mixture of those strong responses and the weak activity. And the basic idea is like the system is not totally linear. So when those kind of for very lower intensity stimuli compared to those typical strong stimuli, we wonder whether it can permit a little bit faster hemodynamic response. And by faster, we basically mean it has a little bit faster time to peak, and also narrower dispersion, basically narrow four-ways of half maximums. So to test this idea, we performed the sensory stimuli. So first is that you can see we use two different visual stimuli at different contrast, and we characterize the response in low intensity and high intensity stimuli. As you can see, for the low intensity stimuli, it really kind of evokes hemodynamic responses with faster time to peak, and also narrower four-ways half maximums compared to the stronger stimuli. So each dotted line here connects the results of a single subject. Sorry. And we also try to tested in different parts of the brain. And here we use a vibro tactile stimuli. So basically there is a vibrant chip attached to the subject's fingers. And then we design the stimuli to be at different levels. And again, you can see for the lowest intensity stimuli, it has a fastest time to peak and also narrower dispersion compared to medium and higher intensity stimuli. So basically, this suggests lower intensity stimuli can evoke faster hemodynamic changes, which may be one candidate mechanisms contributing to those faster changes people see in the resting state condition. But while we provide some possibility, it's feasible to see or capture faster dynamics using fMRI. We also notice like that signal intensity really drops quickly as function of echo time, as shown by the slope of these different frequencies here. And that actually motivates us to think whether some of these faster functional changes arise from data preprocessing instead of biophysical mechanisms. So indeed, we later found like there could be some artifacts introduced to these higher frequency regime that contributing to these functional activations characterized by fMRI. So the idea is actually very simple. So for fMRI data analysis, usually people will do a pre-processing step called nuisance regression. So for instance, this is your fMRI time course. You think this data comprises of interesting neuronal fluctuations in the residual, but also many known nuisance factors such as subject's motion or physiological changes like heart rate or respiration changes. These can be modeled by peripheral measures. And then by modeling fMRI data as a mixture of these things, you just project these nuisance factors out of the data just using a very simple ordinary least square fitting. So this is a very standard denoising step of fMRI. But the caveat here is like, this may introduce frequency crosstalks of your data. So this is just a toy example. Let's imagine your observation doesn't have higher frequency components, only have a slow oscillation at 0.04 hertz. And then in your nuisance factor, you actually have some high frequency components at 0.4 hertz. You can see those high frequency riding on top of the slow oscillations here. And then if you do nuisance regression, here is the residuals of the spectrum of the residual of the residual signals. As you can see, while it kind of denotes well in the low frequency bands, it also introduced a high frequency component here at 0.4 hertz, which doesn't exist in the original data, but in the nuisance factors. So interesting thing here is like, so what do we care first is like, does those nuisance factors in fMRI preprocessing contain high frequency noise? And second, because this magnitude of introduced high frequency component scale positively with the regression parameters of the low frequency signals. So we also wonder whether the regression parameter contain functional information that can propagate to these high frequency parts. So for the first question, actually most nuisance regressors contain high frequency components. So here it just shows the power spectra of some typical nuisance factors for fMRI data analysis. The reason is that lots of measures, they may have white noise in these measures. And also for some motion can have them high frequency components. So that's kind of pose a concern. These nuisance regressors do have these high frequency components. The second question is, does the variance removed by nuisance regression in the low frequency bands contain network structures? Unfortunately, the answer is yes, because there is an early study where they were interesting. They're looking at, typically people look at the brain network structures in the residuals of the data, just clean signal. But in this study, they look at the network structures in the nuisance factors projected out of the data. They can still identify the typical patterns, just mimic those neuronal signals. And then the idea here is also very straightforward, because in the variance removed by nuisance regression, of course, it contains noisy fluctuations. But when you have a lot of regressors, these lots of regressors also constitute a low rank approximation of the high dimensional functional information in the data. So an extreme case is that if the number of regressors equal the number of degrees of freedom of your original data, and all of your functional information will be projected out of the data. So collectively, these mean that even if the data doesn't have high frequency information, post-nuisance regression is going to introduce spurious high-frequency functional connectivity or patterns in the data. So to test these ideas, we use dummy data sets. Basically, we try to remove all those kind of high-frequency information in the data. So what we did is this is a real data. For each brain region, you have its time course, and we take its Fourier transform, so get its power, and then we randomly scramble the faces in the high frequency parts, and then we reverse the Fourier transform back to generate dummy data sets. So for the dummy data set in the low frequency band, it has the same signal structures as the real data, but at higher frequencies, it basically has no correlation with the original real data and has no true activation there. And here we show the results about how nuisance regression can introduce the network structures in the data. So for instance, here it shows one brain slice, and the red region shows regions activated by have functional association. It basically has particular network patterns. And you can see in high frequency bands, it doesn't have any clear network patterns. And then if you do nuisance regression, what I'm showing here, each column is just different combinations of nuisance regressors. And then the direction toward the right direction, meaning there will be increasing number of regressors. So basically how the different combination denoise the data in a way already consistent with previous reports, which I won't go into much details. But if you look at the higher frequency band, even the data originally doesn't have those higher frequency network structures. Post-nuisance regression is going to introduce a lot of network structures here, kind of mimic the lower frequency band information, and those are just purely introduced by nuisance regression. And with the increasing number of regressors and the patterns become more prominent, that makes sense because when you have more regressors, it's going to fit the original data space better. Okay, so to put a short summary of the first section, so we first show that the intrinsic activity can occur at faster temporal scales than previously considered. So it's kind of exciting because it means that fMRI can track brain oscillations above 0.1 Hertz even into the delta wave band. And second, it can means that it can yield cleaner representations of neuroactivity, because in the low frequency band, you have lots of influence of physiological changes, which I will talk in the next section. And there are many potential mechanisms has been proposed by different groups to account for these higher frequency phenomena. And here we just contribute one candidate mechanism that the whole system is not quite linear, and then the lower intensity stimuli can trigger faster hemodynamic changes that support these faster brain oscillations. And third is, while it's kind of promising, there are also potential caveats in data preprocessing that we need to be careful. Otherwise, it may introduce spurious high frequency signals. Okay, so for the second part, I'm going to share how fMRI can image both intrinsic functional changes, but also sensitive to physiological dynamics, and how multimodal imaging, like combining fMRI with PET and EEG, can provide new insights into these phenomena. So as I mentioned, even you are not doing any task, simply look at brain signals across the time, and the red means like positive changes, and the blue means negative changes. You can already see there are lots of dynamics during the session of a scan. And then this is intrinsic dynamics are super interesting for several reasons. First is like it accounts for the majority of cerebral energy expenditure of the brain. So that meaning is really kind of sustain consciousness and then play a key role in maintaining our brain's function. And also, they're not just pure noise. So for instance, if you look at the time course in two remote brain regions, shown these two sites here, you can already see there's some like level of temporal synchrony between these regions. And in this setting, they call it a kind of brain connectivity. Basically, the region have a stronger temporal correlation in their fMRI signals. They are considered to have a higher functional connectivity. This term is more defined in the particular regime of functional neuroimaging, kind of deviating a bit from the traditional neuroscience definitions. So regions kind of having a strong correlation with each other kind of form a network shown by different colors here. And people have identified more than 10 resting state networks kind of operating simultaneously in the brain. While it's kind of super interesting to look at those structured patterns in the functional activity, there are also concerns associated with fMRI, because it's kind of depend on the hemodynamic changes, but like many physiological processes such as breathing changes or heart rate changes can also trigger permanent changes in the fMRI signals. So for instance, here is a subject's respiratory recordings. You can see when the subjects take a deep breath, there is a drastic change in the network patterns. But this is not due to the neuronal changes. It's just like when there's a deep breath, it's going to cause a systemic changes in the fMRI signal everywhere. So it looks like it has a positive correlations between a lot of brain regions. So this is actually has been shown by studies many years ago. The systemic changes in physiology will cause kind of global consistent response in different parts of the brain. And why I became interested was like one day I was examining a data set and then using traditional ways to decompose the data into different components, you can see these typical function network structures. But if you look at the time course associated with each network structure, it looks quite similar to each other. And then if you contrast this time course to these respiratory measures, and it's kind of surprising, they kind of track it quite well. It's kind of interesting. It looks like just those subtle changes in their temporal patterns or its delay already suffice to separate all these brain function information into different networks. So a second thing is it may not be that surprising because our brain vasculature are not homogeneous everywhere. So even if there is a systemic visual input, it may be filtered through these heterogeneous vascular tree and leading to spatially heterogeneous vascular responses. And then, so the question here is that whether these vascular responses may have some structure patterns that resemble those neuronal patterns. So to characterize the region-wise physiological responses, we use a big data set approach and then combined with a linear deconvolution. So the idea here is that for each subject's data, we have this input of respiratory variation or heart rate variability measured by external physio measures. And then here we have voxel-wise fMRI response. And then by linear deconvolution, we can get the response at a single brain region. And we have access to a very large open data sets. So it allows us to look at the region-wise responses and see how different it is across the brain. So what I'm going to show first here is the global fMRI dynamics when there is a positive change in the respiratory variation when you take a deep breath. So it's going to be a 4-dimensional movie showing how when there is an increase in respiratory variation, what's the spatial temporal dynamics of fMRI signals look like. So as you can see, it really has very heterogeneous patterns, first starting in the primary sensory region and later propagating more from the parietal regions. And if you do a spatial clustering to look at the patterns of these 4D dynamics and different colors here highlight different cluster membership, you can already see some structured or symmetric between left and right hemisphere patterns, like mimic those brain network structures. And here it shows the response when there is a decrease in the heart rate. So in this case, probably looks slightly more homogeneous within the gray matter, and the most obvious changes occur between white matter ventricle regions and the gray matter. So, but if you look at into increase the class number to look into the structures in the gray matter, you can still see those very clear network structure patterns here. So this is a bit concerning, that meaning some of these network structures you see by fMRI may not simply arise from neuronal mechanisms. Some may arise from functional mechanisms. So that's kind of general concern of fMRI by many people. It's like, while it has excellent sensitivity and whole brain coverage, it also have a relatively modest neuronal specificity. So to address this limitation, we kind of can think about combining information across different modalities. For instance, for PET imaging, it looks at those brain metabolism and neurochemical processes. It has a more enhanced neuronal specificity compared to fMRI, particularly meaning not sensitive to changes in systemic physiology. And second, it really have access to a broad array of neurochemical processes such as neuromodulators and energy supply that cannot be teased apart by fMRI. And on the other hand, nowadays we have those hybrid PET and MR scanners that integrate these modalities in a single session, and then allowing us to track the dynamic changes in the molecular processes, in the dynamic changes in the functional dynamics. So for the later part, I would just first share a kind of new dynamic PET imaging technique. So for PET FDG, it's a FDA-approved tracer, like to look at these glucose changes in the brain, because when there are neural activity, there will be also glucose changes. So this is also a means for us to track which brain regions are activated by the task. So it was first introduced or tested in about more than 30 years ago now. So basic idea here is that they first do one image and do kinetic modeling to quantify the metabolic levels in the baseline condition. And you do a separate scan when the subject doing a task condition, and then you acquire new metabolic measures. So, and then you take the subtraction of these two. You can add in, oh, this region has enhanced glucose consumption during the task, meaning this region is more related to the specific task. There's many downsides of this approach. Of course, first is radioactive, but on the other hand, it's always in two separate scans. There are like inter-scan variability really kind of incur penalties on the sensitivity and specificity of this technique. So that's why later when fMRI comes out, it really takes over for functional imaging. But a few years ago, like there is a new technique developed at our center, really allow us to look at the dynamic changes within a single session. So I wouldn't go into the mechanisms in much details, but here just illustration how it works. So you have this task going on here, and typically if there is no task in the particular tracer injection paradigm, you will see a signal will continually increasing. When it reaches a steady state, roughly look like a linear line. For instance, in the visual cortex, it really activates the occipital region, not the frontal region. So in the frontal region, it roughly looks like a linear line here. But if you look at the occipital region, every time there is a task, that will be an increase in its slope. So the slope in each condition is roughly proportional to the glucose consumption in that particular state. So it's most obvious when you remove the linear trends. For instance, shown here, as you can see, in the detrended curve, every time there's a task, that will be increased in slope. So likewise, if there's a decrease in metabolism, there will be a reduction in slope. So that'd be kind of exciting. So the idea here is like we can use this probably to classify whether some sources we see in fMRI signals are neural or physiological. And then the particular case we are testing is sleep. Why this is interesting is like when people fall asleep, shown by EEG case here, there will be more coupled fMRI signal with systemic physiology here. So for instance, this is a global fMRI signal going later on. And then these are the respiratory measures. As you can see during the sleep drowsy cases, there are more accentuated anti-correlations between these two measures compared to resting state conditions, awake conditions. So the basic idea is that we may collect simultaneous fMRI and PET changes during wake and sleep. The hypothesis is that if it's neuronal mechanisms or some regions, you can see it in PET dynamics as well. If it's more in neuronal physiological mechanisms, then meaning we may not see much in the PET dynamics. So here's the acquisition protocol. We basically have simultaneous EEG and the PET and MR scan over 90 minutes, and then the subjects allowed to fall asleep during the entire scan. And this is just an illustration of how this paradigm works. So for instance, here is the EEG measure. We use that to indicate when the subject is awake and sleep. And this is a standard spectrogram of EEG electrodes in the occipital region. As you can see, when people have this delta alpha wave bands in the 8 to 12 hertz oscillations, the subject is awake. And when they sleep, it will be replaced by lower frequency oscillations. And then in this particular subject, they have wake, sleep, wake, and sleep over a 90-minute scan. And if you look at the fMRI signals, you can see a drastic contrast between sleep and awake. I won't go into much details. I know it's a bit counterintuitive. There is increased fMRI during sleep. There are many hypotheses behind it. I'm happy to talk about it if you're interested. And then if you look at PET dynamics, apparently when people fall asleep, you will see a decrease in its slope, meaning reduction in metabolism. And when subject become awake, there is a slight increase in metabolism. And when subject fall asleep, there is decrease in metabolism again. And here is another subject example where you have different stages of sleep during scan. And you can clearly see in each stage, there is a different slope of the signals. So here we characterize the spatial patterns just to compare them. For fMRI signal, we calculate the fractional increase in the fMRI fluctuations, and this shows the group level responses of which region shows the most drastic changes. And here we quantify this PET metabolism, and here shows regions shows the most prominent decrease in these glucose metabolisms. So you can roughly see there are regions kind of very complementary to each other, don't overlap that much. So for here, just like show those cortical patterns to make it more clear comparisons, kind of rendered on the surface view. As you can see, if you plot these patches of hemodynamic changes and metabolic changes together, looks like they're positive aligned, but because metabolism is reduction and hemodynamic signals increase, they're actually quite complementary to each other, as shown by regions have the strongest changes here. So we kind of maybe interpret more liking they have different mechanisms. So for in gray matter, most of the energy is kind of dominated by signaling. So that's why you look at this glucose metabolism, those regions are most active during the task condition. They really have a decrease in metabolism. But for the fMRI signal, as you can see, the regions shows the most prominent changes are in the sensory region. It's quite different from these PET measures. And now we probably more attribute to these physiological dynamics, because that pattern is also quite similar to the previous respiratory changes coupled patterns I show in the physio network cases. So collectively, one potential interpretation of these fMRI signals, we do think it has a considerable physiological fluctuations instead of local neuronal changes to the characterized patterns. And owing to the sensitivity of our technique, we also use that to look at white matter changes. But due to that considerations of time, I wouldn't go into much details into it. I just like quickly showing that it's also possible to see sleep-induced changes in these white matter signals. And the polarity is very similar to gray matter, but just to a reduced level. And we're also looking into different tracks within the gray matter. Oh, in the white matter, you can see alignment between metabolic and hemodynamic fluctuations. So there are kind of several mechanisms relating to it, and again, I wouldn't go into much details, but basically we're considering there is a link between the different tracks had different vascular density that are linking these hemodynamic and metabolic signals. Okay, so a summary of the second section. I first show that fMRI can be used to extract rich information in both functional and physiological dynamics, and then these physio-coupled fMRI dynamics can exhibit structure patterns that mimic large-scale neuronal changes. And then I show like by combining fMRI with PET and EEG, it may provide a means to disentangle or get some insights about the relative contribution of neural versus physiological fluctuations. And we check those changes in the non-REM sleep cases. First, we show in gray matter, regions shows the most prominent changes are very complementary to each other. But in the white matter, they have a different pattern. They are more have a modest consistency of distributions. And for gray and white matter, they have quite distinct physiological mechanisms and also metabolic consumptions. As I showed in the previous slides, they may arise have different mechanisms. We can, if you're interested, I'm happy to share further about our thoughts on it. So yeah, I hope like this just give you some flavors of what my lab is interested in doing. On one hand, we are interested to addressing this limitation of different techniques and also to combining them to devise new means to look at the brain's function and physiology. And on the other hand, we're also interested in applying them to provide new insights of these functional and physiodynamics in both healthy subjects and diseased populations. Okay, so finally, I want to acknowledge all my mentors during my postdoc and also during my PhD training and all my colleagues and show in these slides, and also NIH funding can support all this research. And just put a small like advertisements as I'm starting my own lab, we're also hiring postdocs and grad students. So if you're interested in joining us, or if you're interested to do neuroimaging, feel free just to reach out. Okay, yeah, I think it's about time. So I will just end here. I'm happy to take any questions now or later on after the second talk. Thanks.